Thank you so much. Thanks for having me, everybody. And I'm excited to talk about InflammaSense today. So InflammaSense is building sensors to identify clinical states that may be responsive to therapies, right? And this can be used in the clinical setting, such as a hospital, or it can be used in clinical trials. Our first use case scenario is sepsis. And so the problem with sepsis is that every hour you don't identify it in the hospital, there's a 7% mortality risk increase, okay? So every hour can save that patient's lives if you identify it early. Every hospital in the US loses $2.2 million per year for sepsis losses. It's the most expensive CPT code, and it's the most expensive patient that hospital sees, right? The reason why it's so expensive is that those patients get readmitted, right? Because they leave the hospital, and if they get readmitted with the same diagnosis, the hospital has to pay for that readmission, okay? So do clinicians care? They do, but administrators of the hospital care even more, right? Because this is such a big impact on their bottom line. It's also important because those patients that come back, 50% of them are going to die, right? So those value-based payments that the hospital is laser focused on is decreasing due to these patients that are readmitted and then subsequently die. The ICU docs care because these patients, we can't treat them, right? So they're essentially doing poorly and there's not much we can do. There are current systems out there currently that evaluate the electronic medical record, right? And so we build AI models where we can identify if somebody is gonna get sick or have severe sepsis. But the problem with these models is that they're relegated to the interval at which you obtain those vital signs every 2 hours, every 6 hours, every 12 hours, okay? So no model can perform at high accuracy or area under the curve beyond a 6-hour horizon. So 6 hours before the clinician identifies that patient has sepsis. So therefore, they're low accuracy. What's the difference? So what are we doing and what's our solution and value proposition? Essentially, we record 14,000 times per second neural data from the ventral cervical area. The vagus nerve and other sympathetic nerves respond to inflammatory molecules called cytokines, which increase in sepsis. We've been funded through BARDA and run 45 patients so far, where we've used a sepsis model to identify those inflammatory cytokines with our neural sensor. In addition to that, we also run an EMR model. So always running is an EMR model that identifies whether or not somebody is going to go south based on vital signs and laboratory values. Our novel sensor, again, is an array that's placed in this real estate over the neck, measures neural firing and every single vital sign you can think of at the bedside that you would obtain from the ICU. The concatenated model brings together the EMR data plus our neural data. And as soon as somebody has an indication that from our LLMs that are monitoring the data from the EMR, if somebody has a presumed infectious diagnosis, we then signal to the nursing staff to place our sensor on the patient. And within 5 minutes, we can get an idea of where that ramp up is. How quickly is the neural firing changing in accordance with the cytokine concentration? This is just some of the data for 2 different cytokines, TNF and IL-8. And you can see that there is a huge heterogeneity. If I took everybody in this room and I gave you an infection with E. coli, there would be huge differences between everybody in this room, right? So it's important to know who's going to be sicker and who's going to be less sick and how we may treat that patient. The patents that we have are 1 is granted, 2 are pending, 3 are in process. The business model is essentially $400K per year. Again, it's 2.2 million that they're losing per year on sepsis cases. If we can reduce the hospital stay by reducing those readmissions, then we can subsequently decrease the losses that are due to these sepsis patients. We expect to reduce it by 1.1 million per year. The total addressable market is 13.6. The TAM is 680, and we expect within 2 years to get into 20 hospitals with a return of 8 million. Other companies, Edward Life Sciences, just was acquired. Their sepsis monitoring unit, which was a bedside sepsis device, was acquired by BD for 4.9 million, and Rothman Index was also acquired by BD. So this is our development plan in full, and essentially we're here. We've got angel investment already. We have government funding as well as some venture capital pre-seed funding, and we're looking for a seed to essentially develop our MVPs, obtain 510K, and develop what's called a federated learning platform, so we can take those models and federate them across different hospitals. This will generate data that's necessary for the feasibility, the pivotal trial, and to get FDA clearance. In the interim, however, we are going to be focusing on vital sign monitoring, right, for clinical trials. Right? I mentioned clinical trials as well as in the hospital. And the goal is to understand, can we identify patients that may be responders or non-responders to a specific therapy based upon this inflammatory cascade? The use of funds is mostly going to be used for salaries, but also for IP and engineering and regulatory. As I mentioned, this is where we are. We have funding for the green, dark green bars here. We have new funding where we're developing a closed loop device. I was excited to see that there was interest in the closed loop this morning. And that Manfred mentioned actually that there is this idea that there are certain aspects of closed loop that were that coalesce into one thing. So inflammation can lower the threshold for pain. Inflammation is also very much core to mental health disorders. So this is the project that we have funded through DARPA and ARPA to build a closed loop device to monitor these neural signals, to titrate the amount of focused ultrasound to the spleen to inhibit inflammation. The other part that we're working on is mental health, right? So I mentioned inflammation is core to sepsis. Inflammation in the brain is also core to severe mental health disorder. And you may say, like, what? How? How does that even work? Like, what is going on? Right? So the model that we've developed is we inject humans with something called LPS, which is a E. coli sugar lipid epitope. Everybody in this room has LPS binding protein circulating in their blood. And the reason why is because their E. coli in your gut can leak out, and your LPS binding protein binds to it, so it doesn't cause a huge inflammatory reaction. Actually, in Alzheimer's and autoimmune disorders, there's increased LPS binding protein circulating in those disease disorders. So the question is, can I take a look at your response to this inflammatory molecule and identify whether or not you are going to have mental health disorder and the severity of that mental health disorder? The model that we do is, again, we inject these humans with LPS, and then we monitor their inflammation as well as their symptoms. But we also have different patients. Like I said, everybody's different in this room. There's also differences in our mental health, right? So across the continuum, you may be a little bit anxious or you may be severely anxious, right? So understanding where you sit on that continuum to be able to provide you with the correct therapy. So what's the state of the art? So this is the marine resiliency study that was run out of UCSD, or part of our neuroscience unit. And it's 5,270 soldiers at Camp Pendleton that were monitored before they were ever sent to war, right? So the question is, can we identify patients or soldiers who are at risk for mental health disorder before they're ever sent out? And the answer is, indeed, this is part of the multiple different papers for this marine resiliency study, around 40 that are out there. But bottom line is the soldiers who are more inflamed, as well as who have low vagal tone, are more likely to develop PTSD, okay? So this is before they were ever, these are seemingly healthy soldiers that essentially were monitored before they went to war. Sometimes they had 2 deployments, and when they came back, we found that the guys who had PTSD had this preemptive inflammatory cascade. And this has been borne out in multiple, it's not just PTSD, depression, anxiety. We see that there's very much a coalescing of inflammatory signaling that results in release of more stress hormones and more sympathetic output that desensitizes these immune cells to the usual anti-inflammatory effects, right, of vagal output, right? So we differentiate these patients into endotypes, either high or low. sympathetic drive, where you can have changes in the beta-2 adrenergic receptor, as well as this alpha-7 nicotinic acetylcholinergic receptor. When you look at in the tube, if I just take your blood and I add LPS to that tube, I can look at how much inflammation will occur when I add LPS, so how much TNF is released. If I add an agonist to the beta-2 adrenergic receptor, I can also see, okay, how does this anti-inflammatory pathway work? Is it capable of decreasing inflammation in this patient population? And it turns out in obesity, overweight, as well as PTSD and depression, the amount of inhibition is decreased for the beta-2 adrenergic receptor, as well as for the alpha-7. So I'm going into the molecular dynamics, but is there a way we can connect this neural signaling to the molecular dynamics that encode inflammation in humans? And so we did this work, it's now published in Nature Scientific Reports. You take healthy and seemingly healthy individuals, we have them go through a cold pressor test, this stress test. We talked about different biomarkers for pain earlier this morning. The cold pressor test is a painful test, but it also initiates an autonomic system response, right? And so what we found is that the different features in our autonomic system that measures neural firing as well as cardiac rate can predict the amount of cytokine somebody will have after I give them LPS. So this is before they ever got LPS. If I took you in this room and had you stick your hand into a cold ice water bucket, I can get a prediction of the amount of inflammation that you may have if you did have an inflammatory cascade occur. Similarly, across another autonomic test, this is a deep respiration test. We have patients, or just subjects, healthy subjects, do deep breathing up and down for 20 breaths, and they segregate based upon their autonomic response. And finally, we looked at just the mental, they're, again, seemingly healthy individuals, but some of them did have PTSD, depression, and anxiety on their scores, right? So we looked at their neural firing, and we correlate that to their symptom scores for each of these mental health disorders, and we found very high correlations and very high effect sizes and power. So for this particular vertical, for the mental health component, is that can I identify different components within each individual and say, look, are you going to be a responder to this particular drug or not, right? Or are you too severe for this particular drug trial? So can we decrease the non-responders within clinical trials? And so we've been having some nice traction with this idea. The markets are here, and the amount that we would charge is $375,000 per trial. The goal is to have a faster trial, improve trial success, and better data quality and outcomes. There's a lot of people that are in this space. There's been some recent failures. Altos just had a failure in the last month. But we think that our particular solution is better, and we've seen it bear out in our trials. So again, can we identify these neural signatures of response that may identify non-responders for drugs? Our regulatory pathway is, again, to identify, to get approval for our vital sign monitoring component first, and then move it to a de novo application incorporating our other components. We have multiple different sources of funding, and we have pre-seed funding for 250. We're looking for a 250K from Kitty Hawk. We're looking for a lead for our seed round, which is scoped at 2.9. And so this is our team that's across UCSD and InflammaSense. And with that, I want to say thank you, and I'll take questions.
Okay, do we have questions for Dr.? I see we have a couple.
Is it possible that you have co-founding factors that you have inflammation where you can get false positives?
So we've looked at false positive rates, and we also look at false positive rates in our EMR modeling, not only our sensor, right? So we want to lower those as much as possible, and we've done that across. We have 13 million patient encounters through the UC-wide system that we've run our model on. So we've minimized those over iterative algorithmic building that we've done, different algorithms that we built. But for sure, you can imagine that any kind of stress response, right? Again, we talked about acute pain earlier this morning when I said there's, the InflammaSense, there's the Sandia Subramanian's work where you're looking at acute pain in a operative setting, right? Any kind of stress can cause an autonomic response. Our goal is to induce an autonomic response, right, with our autonomic tests that then predict your likelihood of inflammation. So we're seeing very good results with that, and I'd say, and they're published, so you can look them up in Nature Communication Biology.
On the topic of mental health, two questions. One is, your data on the predictive component of the what is it, the LPS? Is that predictive or correlative? That's number one.
It's quantitative linear. So we have linear regressions, multivariate linear regressions that use different features that I can't really talk about. But bottom line is that we do have a capability to, within a 5 picogram per mil of TNF granularity, predict where somebody's peak inflammatory response would be, as well as the likelihood in quartiles or multi-class classifiers that we have for 3 levels. So we have different, we have capability to run the regressions, run our machine and deep learning classifiers to identify these different levels.
Okay. And then the second question is, now what after you've identified that these people are at risk for like a mood disorder like PTSD or depression? Typically these people have to be triggered to really move into a pathological state. So what is the end goal of this?
So we're running Vegas. We do a bunch of different neuromodulation trials, and we're also looking at, the standard that's out there. So TMS, ECT, and ketamine, actually. So we're looking at patients before they get any of those treatments, and then seeing if we can predict responsivity to the therapy, where we think some of these therapies may have, effects on the immune system within the brain. So it's not only the neuroplasticity, but we think there's neuroimmunoplasticity, and maybe even more so with these psychedelic drugs like MDMA and the psilocybin. But the goal is, can we identify, okay, are there different groups of patients that may be more apt to be a responder to that said therapy, or should you change your algorithm so that this person is going to ECT first, or this person is going to TMS then? So the algorithmic approach of how you may treat these patients based upon a more granular look at their mental health severity may help you get to a better treatment faster.
Okay, any other questions?
That was interesting. When you're measuring the vagal nerve, you're measuring a response over time, right? So cytokine concentrations are variant and they have different time kinetics, TNF, TGF, all these things are changing at different times. How long do you have to measure before your signal is robust?
So this is the raw data, and basically we get, with 5 minutes of recording, we can start to see neural firing changes, and there's also differences in geography, right? So nodus ganglion has much larger, like this area has larger amplitude, as well as higher. It's a code multiplexer, right? So you have 4 action potentials that turns into 20. So we see increased frequency and amplitude in different areas where we measure. Yeah.
Now, if you're doing these, you're not catching the changes on the cytokines.
We're doing cytokine measurements.
So this is changing on a 5-minute time frame?
Oh, so we do every 30 minutes cytokine.
Got it.
Yeah. During the LPS trial, we did every 30 minutes. In the hospital we're running right now, we're doing twice daily or 3 times daily.
Okay. Well, thank you very much, Dr. Lemon. Excellent presentation.